Hi everyone, I'm Haley. I'm a PhD student here in the biology department, and I'm going to be showing you around the research greenhouse today.
The research greenhouse is where many labs on campus conduct their evolutionary or ecological research, particularly those that require plant material. The building was completed in 1996 and has 7 different spaces shared among several labs for growing plants, along with storage, preparation, and research space. Access is restricted to researchers caring for their experimental populations. Each room's temperature and lighting are independently controlled via computer to be able to provide the best growing conditions for its inhabitants. In summer, it can be difficult to regulate heat and humidity, so shade cloth moves across the ceiling panes throughout the day to help regulate temperature. We also use an evaporative cooling system, which releases water into the air. When water evaporates, it absorbs heat energy to change from a liquid to a gas, meaning it's a cooling process. It can get so hot in the summer that we sometimes need an additional swamp cooler to help out the innate systems. Plants are watered regularly by a combination of faculty, graduate student researchers, and trained undergraduate researchers. Each type of plant has very specific watering requirements, so it can be disastrous if they get too much or too little water. One of the issues that can arise if plants are overwatered is they are unable to take up all of the water that they're given, leaving plants to sit in stagnant water. When this happens, it can attract pests, like woolly aphids or agalids. Pesticides are avoided in the greenhouse except as a last resort, and biological controls are used instead. Usually, this means that when we notice a pest problem, we release a benign predator insect like ladybugs or lacewing flies, which hatch out of these cardboard shelters and eat the pest insects. Now let's explore some of the research rooms. This one here is occupied by the Sobel Lab, which does research on the formation of species. We have a few species of the genus Mimulus in here, which are the monkey flowers. We have some desert species in here that are native to California, such as Mimulus whitneyi, and these purple ones here are Mimulus ringens, and they are native to this area and grow on riverbanks. One of the large-scale projects the Sobel Lab is participating in is a national collaboration with the Joint Genome Institute, which is working to assemble the genomes of several species. With a project this big, the lab accepts undergraduate research assistants to help collect data. Caroline Gilmore is overseeing some here.
What are we sensitive about? Time. Very good. Okay. So we flash for instance. Good. Okay. Right now we are collecting tissue via liquid nitrogen collections. We are flash freezing it for future genomic work, so we're going to be doing DNA and RNA extractions on them. We are also, on a couple select plants in here, going to be collecting tissue for a JGI grant in which we flash freeze the tissue and send it off to JGI and Hudson Alpha for sequencing to be aligned in a phylogenetic analysis with a few dozen other species. We're really excited to be in part of this project with them.
DNA begins to degrade as soon as tissue is removed from the plant, so to make sure it stays intact for the downstream analyses and sequencing, we quickly dip the tissue into liquid nitrogen to prevent decay, and then store samples at -80 degrees Celsius. Samples have to be shipped with dry ice to make sure they stay cold. This space is used by a few different classes and labs, including Dr. Jessica Hua's lab and the plant ecology course. In a collaboration with Dr. Hua, the plant ecology class is working on a project to investigate how aquatic plants uptake copper, which is a common part of pesticides used in farming. These kiddie pools may look out of place, but they are actually being used as mesocosms. experimental environments that mimic natural environments. These are being used to investigate if some communities of hosts are more susceptible to disease than others. This space is used by the Ecological Genetics stream of the Freshman Research Immersion Program. This stream uses both plants and insects to answer ecological questions, such as how environmental toxicants can impact growth and development, and specifically focuses on the genetics of adaptive change. Veil cress is one of the most commonly used to study species because of its fully sequenced genome. Another one of the main spaces in the research greenhouse is the head house. This is shared space between researchers, and this workbench here is often used for projects and studies that require more space than is usually available in the propagation rooms. So on this space, we'll normally do transplants of our plants, we'll collect plant material or tissue for further study, and we'll also do some trait measuring here. And here we have Hoger doing some of her research for her project. Hoger, can you explain to us what you're doing today?
Hi, my name is Hoger. I'm a master's student at the Silver Lab, and what I'm doing here is basically process or artificial process between two different species of Mimulus. So here I have Mimulus whitneyi, and those are Mimulus constrictus. And the reason why I'm doing that is because in nature, usually when two different species interbreed together, they will produce a viable hybrid, meaning this hybrid will suffer a reduction in fitness. I want to see if this is happening between the two species that I have here. And this will ultimately help to answer the question, what is maintaining two species separate and why they have reproductive isolation, and kind of explain the secret of biodiversity on our planet. And the reason why I'm using dissecting microscope is, as you can see here, my flowers are really, really small, and the compartments inside of it are even smaller. So I use the microscope to look at the anther, which is the male part of a flower, ensure the pollen is enough and in a good quality. So when I move it to a female part of another flower, I ensure a good quality cross.
This room is also used by the Sobel Lab and houses more plants from the genus Mimulus, section Diplochus. There are currently 8 species up here, all from chaparral environments. These are habitats in typically the western United States and have a Mediterranean climate with hot, dry summers and mild, damp winters. They are often considered to be biodiversity hotspots. These species are being used in a large-scale experiment looking into intrinsic barriers to hybridization among species, which can manifest as malformed hybrid seeds. Plants of the same species from different populations and plants from different species are cross-fertilized. The inability to produce healthy hybrid seeds can strengthen the divergence into different species. To see how these are measured, let's go to the lower greenhouse prep area. One of the most important spaces in the prep area is the phenotyping room, where we measure and collect data on a plant's phenotype, or trait values. When we have a lot of plants to process, often the most efficient way to measure them is to take photos, and then digitally measure the trait on the photos. Here we have Irene Martinez taking photos of the seeds of the hybrid crosses from the plants upstairs.
So here we have our seed morphology station, where we look to assess the ratio between good seeds versus bad seeds and empty ovules. So here you can see we have our light box set up and a camera, and what we're doing is we're actually taking pictures of seeds in order to see whether they're good, whether they're not good, or whether they're empty ovules. So you can see here we have what looks like a good seed. Next to it we have a little bit more shriveled seed, so we're going to assume that it's inviable, but we won't know until later. And then we have some empty ovules here that haven't been fertilized.
Comparing the ratio of healthy to unhealthy hybrid seeds tells how strong the barriers to reproduction are between the species.
And what will you do to test if the seeds are inviable later?
We will do a tetrazolium assay where the seeds will actually stain bright red that are alive, and the inviable ones will actually not stain at all.
A tetrazolium assay is a biochemical test that determines if seeds are actually alive, even if they're dormant, based on the activity of respiration in the seed. When seeds are hydrated with tetrazolium, a living seed will release hydrogen ions, which interacts with the chemical to stain the living cells red. The prep area is also where we prepare soil for planting, store materials, and clean pots. Often the species we use will require a specific mix of nutrients and components, so we mix those here. For example, a species we're using currently is specialized to limestone soil because of the high pH, so we have limestone to mix in, and we have peat for when we need things to be more acidic. Here is where water is treated. The water is put through a reverse osmosis filter to remove contaminants and make sure the plants are not exposed to excess minerals that would affect their growth. As you can see, it takes a lot of effort to properly care for and collect data from plants. If you want to get involved with a research project, reach out to one of the labs mentioned in this video and see if they need your help. Thanks for watching!